The first one is the development of a new mouse model for PWS. And this is from Dr. Muscatelli and Dr. Boretz, who are in France and who have worked collaboratively for a number of years. Dr. Muscatelli has expertise in PWS mouse models. She's done a lot of the work looking at, or that initial work that showed that oxytocin in the newborn period can correct a lot of deficiencies in PWS in a mouse model. And she has worked with Dr. Boret, who is a neurobiologist who looks at circuitry in the brain. So it's a really nice collaboration. And they've done a lot of really interesting work to understand how the brain development is different in PWS compared to typical, well, in this case, mice. An issue with the PWS mice is if we knock out that entire region as it is in humans, the mice typically don't live. And so the field has taken to deleting one gene at a time. And that is valuable and tells you about each gene, but it leaves something to be desired because some of these genes are related. And so when you knock out one and leave the other, the other gene can compensate for the one that's been knocked out. So in this case, Dr. Muscatelli developed a mouse model in which nectin and MAGL2, which are two related genes that sit right next to each other in the critical region, are deleted at the same time. And they're going to be characterizing the behavioral, developmental, and metabolic characteristics of these animals. So they've newly developed this animal model, and now we'll be doing all of the characterization. So we're excited about this study because this is a team that we know works really well together. They get things done. They understand PWS. And so there's a high likelihood that they'll gain new information from this double knockout. And the other thing is that, all of our mouse models with PWS, they provide us good information, but there are some limitations. And the thought is with this mouse model that is knocking out two related genes at the same time, it may be a more relevant model for testing new therapeutics. So the new mouse model is expected to be more relevant to understanding how the brain develops and how the behaviors are different in PWS, and also, again, provide a model for testing new therapeutics.